I'm sure you're all familiar with the fact that majority of the drugs which are very popular a few years ago are being banned by FDA. To give you an example, Celebrex and Vioxx are drugs which are considered really excellent drugs for inhibiting specific types of molecules. And they did a great job. They did exactly what they were designed to do. But what people didn't understand or realize was that they did much more than what they were supposed to do. This is what systems biology brings to bear on medicine.
Shankar and his students and colleagues have developed some of the most valuable computational infrastructure for bringing together and integrating complex, diverse biological data into useful information for biological scientists to discover new ideas and test their hypotheses.
My research can be broadly categorized as being systems biology and engineering. The goal of the research is to understand how living systems, especially we work on cells, function based on our ability to make large-scale and high-throughput measurements on these systems. And so we can decipher what is the circuitry behind the functioning of these cells, and can we build quantitative models to try to understand whether we can predict phenotypes from understanding individual parts of the cell.
What particularly sets Shankar apart in this field is that in addition to deploying these valuable resources to the community, he also has taken that integrated information and used it to develop novel engineering models that can predict the function of signaling pathways in cells and other complex biological processes. Then he takes the predictions of those models and uses them to team up with experimental scientists to generate new experimentally testable hypotheses, gain new insights into biological mechanisms and disease processes.
Can we identify therapeutic targets, design drugs that would help us not just fight a disease at the local level, but the level of a whole system?
Shankar has focused a substantial part of his research effort on complex human diseases like diabetes, and these are really the hard problems to solve in systems biology, but they're also the ones where systems biology has potentially the greatest impact on human health and well-being.
We can build a picture of these cells at the level of the whole system, where we can ask, what does the drug do in addition to doing what it is targeted for? And this will help us find the pros and cons of using these drugs. Now, this also extends to areas like regenerative medicine, where we ask the question whether we can understand the circuitry of cells which differentiate from adult stem cells or embryonic stem cells into specific lineages like the heart, the liver, the kidney, etc. So this is where the research is going in the future in terms of analyzing, deconvoluting what the system is, reconstructing models of the system, being able to build quantitative understanding of physiology. The availability of these vast amounts of data and the challenge of transforming this data into knowledge that will give us deep insights into function and that will provide us with the tools for next generation biomedicine is what drives a lot of us towards these directions.